Hello, everyone who's joining for today's session. My name is Modeline Longjohn. I'm a graduate student in the Department of Biochemistry at MON, and today I am so excited to be sharing with you how to isolate DNA from strawberries. Now, it's the same basic principle that you would use in the lab, right? But just you're going to use different equivalents of what we're using today, okay? So in the lab, you could isolate DNA from, let's say, cells that you've grown for a while, or from tissue from, let's say, an animal cell like mice or rat or something like that, but we're not going to do that. That's above our pay grade at this point. But today we'll be isolating DNA from strawberries. So this is something you can find from your house, like anything. You can actually isolate from spinach. I've done that before. That was really fun and cool. I had so much green liquid everywhere. You can do from bananas, you can do from kiwi, all different fruits. So much fun. It's the same basic idea. So the first thing you must have is your fruit, in this case strawberries, right? So I have 3. You can see here that it's thawed. It's not as frozen as it was a few hours ago. I decided to take a nap because I was exhausted. And while I took that nap, I knew it would thaw and be ready for me to do the isolation really quickly. So first thing is your fruit. That's on the side. The next thing is going to be the magic liquid, but not so magic. It's like it contains household stuff. So you've got you're going to have this plastic container combined different ingredients. First thing is clear detergent, right? So you're washing up liquid at home. There's that here, and then you have some salt added, and then you have water, and then you mix that together, and bingo, you have your DNA extraction liquid, right? This is what's going to do the magic. Well, not so much magic, but you're going to see what will happen. And the final thing the next thing we have is isopropanol, right? So this is also rubbing alcohol. We're going to put that in, and you will see the extraction process pulling out the DNA from the whole mashed up strawberry mix, right? So strawberry, I'll show you again, strawberry, DNA extraction liquid, rubbing alcohol, and there is also coffee filter, right? You would use this to drain out you use the coffee filter to drain out the liquid when you've mashed this up. So we're just gonna dive right in and start with our DNA extraction. I'm so excited. Okay, I hope you're as excited as I am. I love all things genetics, DNA, RNA, proteins, their interactions. It's so much fun. You will definitely learn a lot about this if you ever choose to come to MON or come to the university. It's so much fun. I've been learning this for years and I love it. Right, so first step, we would take the DNA and we're going to do a little mash-up job, okay? So I would mash this up. The nicer you mash it up, the better, because what you're doing is that you're breaking it down so you're breaking down from the blob of cells. I'm sure you must have seen the video I put where I said living organisms are a blob of cells, right? Lots of cells make up tissues, right? So we're trying to break this down into as basic a form as possible, and so that would allow us to pick out or get out the DNA. So mash it as much as you can for about 2 minutes. Well, I'm not going to smash it because I don't want to make a mess. So I'm just going to mash it as gently, but you can smash it if you like. I just don't want to make a mess, but if you do, that's fine. Okay, so does this look mashed up enough? I think it does. Oh no, I can still see some particles. So more mashing up, more and more. I think this looks pretty good, right? I think this looks good. Okay, yep. I have mashed it up nicely without smashing, but you can smash yours. Yep. So this is all set, right? This is all set. I've smashed the strawberries into this beautiful paste. I wish I could eat it, but I can't. It's for the experiment. Okay, so now that this is done, the next thing I'm going to add is going to be my DNA extraction liquid. So this is the mixture that contains water, salt, and detergent, right? Your clear detergent at home. Okay, so what I'm going to do, I will add I'm supposed to add 2 tablespoons of this into the bag. Okay, I'm probably just going to get a spoon. So I have my spoon, and I will open up the bag. By the way, I'm using kitchen gloves. You could just use any kind of simple gloves you find at home. That's fine. Right, so I have that, and then I have my DNA extraction liquid. I will take two teaspoons or 2 yeah. I have to follow the protocol. In science, you always have to follow the protocol. It says add 2 teaspoons, and I've got a huge spoon. So I'm gonna get a different spoon. I can use my household spoons because it's safe, right? This is just salt, water, and clear washing liquid, right? So this is 1, and it goes in my bag, and then a second spoon into my bag. Right, okay, you know what, I'm feeling generous, so I'm feeling generous, and I had a lot of strawberries, right? So you have to always think about the proportion of strawberries you have to extraction liquid, right? So I'm feeling generous, and I had a lot of strawberries, so I'll add that, and I'll seal that up. So you would figure out now that it doesn't matter too much if you add too much, right? Okay, so let's see. Okay, I have a lot more strawberries than I intended, so I'm going to add a bit more because I need to be able to see the soap suds. I need to be able to see that I'm making a bit of have soap bubbles in here, and I'm not seeing that. That's probably because I had a lot. I had 3 huge strawberries, right? Okay, so put more. Yeah, I think I got it now. I got enough. Okay, so can you see the soap bubbles? So I can see the soap bubbles. Reseal the bag. Try not to make too many soap bubbles, by the way. So reseal the bag and then smash it up a bit. You can see the soap bubbles. That means you're in the right place. You're doing the right thing. Yeah. So I will do this for 1 more minute. Mix it up nicely for 1 more minute. I think my minute is up now. So then comes the time to do the sieving, right? So I'll take an empty container. I will put my doesn't matter too much, so I'll take this off from one hand so I can bring this out peacefully, right? I'll take this and then I'll put this here, right? Okay, this is going to be the messy part, right? Okay, so put this way, and then I put this aside, and I start to pour it in and see what I get. So that's the coffee filter, and this is the liquid that contains the DNA extraction liquid and the strawberries that I mixed, right, and mashed up. Okay, so nicely, I will try to pour this in and see what I get, right? Okay, this is going good. Okay, right. I don't know how much liquid is coming out. I am not sure. Oh, okay. Oh yeah, it's coming out in bits and pieces. Oops, Anyway, it's coming out slowly. Slowly. So I got a very little. Well, don't be worried if you're making a mess. I'm certainly making a bit of a mess here, but that's fine. Okay, so I'll take another set of coffee filters, and then I'll pour in some more. And see how it goes. It'll be nicer if you had a bigger coffee filter. That would help, right? Because then you can get a lot out really quickly. But the coffee filter I have is tiny, but that's not a problem, right? It's still coming out. I just need to separate the liquid out from the not so liquid part. Okay, so I'll have to get more coffee filters. I have a lot of that, so that's fine. You could actually also do this with a nice little dishcloth, but I'm not sure I want to ruin my dishcloth here. Not ruin per se, but I'll stick to the coffee filter. So I will add some more because I want to get a good amount so I can get out some good amount of DNA from this. So okay, let's see how much I can get out of this. Okay. Okay, so this doesn't look bad. I've got a good enough volume, right? So I'm not going to try to sieve everything because it will be here for the whole night or the whole day per se. So now that I have this, what I'm going to do is I will then pour some of the isopropanol, right? So this is the rubbing alcohol that I had, and then I will pour it down the side slowly so that I get about the same. So I get about twice. I double the volume, right? So if you look here, you can see a clear white cloudy substance. That's the DNA that is forming at the top, right? So in order to pick that out, I will tilt the container to the side. I'll take this off at this point. You can see it's all filled with strawberries. I'll tilt this to the side, and behold, I've isolated DNA from strawberries. That's the DNA, right? DNA is usually it's this entanglement of nucleic acid, and it is see how much DNA I got out of the little strawberry mix that I put in here. So above the clear liquid, you can see just in that interface between the darker liquid and the top part is where I got my strawberries from. So I have isolated DNA from strawberries. You can see I did not isolate a lot. You could isolate a lot or as much as you like, but that's definitely something. So this is how you isolate DNA from fruit. So you first mash it up, and then you add the DNA extraction liquid, and then you sieve it so you get the juice out, and then you add the rubbing alcohol or isopropanol. I know this sounds so easy to me, the way I'm saying it, but I'm sure you can do it. It's really easy. Thank you very much for joining me. Hopefully the next session is fun, and hopefully this works out. Thank you.